Okay, so our next speaker is Krastan Blagoev from the National Science Foundation and Johns Hopkins.
Okay, so...
and his talk will be on nucleotide excision. Why don't you just take it away, please?
Okay. Well, do I need the microphone? Probably not. I want to thank the organizers for inviting me. Thank you very much for the invitation. And do I need a microphone, maybe? No. Okay. If you can hear me. This is a fantastic meeting. The local organizers here, they spend a year and a half working with all sorts of different obstacles. And, well, at the end of the day, we couldn't solve the obstacle of using different computers. Perhaps next year, or even maybe tomorrow and Friday, if we could actually have the presentations on a USB stick, would be great, because one computer works, and so let's use it. Just hard to connect all sorts of. Apparently, 100 years of computer development was not enough. Today I want to talk, I was wondering what to talk about today, but now that everybody's talking about these phase transitions and condensations, and they call them whatsoever, I think that the assembly of machines that are membraneless in the cell is an extremely exciting thing. And the recognition in recent years that these machines on demand that assemble when they're needed, and then the parts are actually recycled after that, is remarkable engineering which we have never envisioned in our engineering efforts on Earth. So I think we can learn a lot from biology, how it assembles these machines, which are very complicated complexes, sometimes involving hundreds of proteins, to do a specific function which is needed at a given point, and then disassembled, and the proteins and the machinery is actually reused. Extremely efficient way of dealing with damage. So today I'm going to talk about one of the pathways of DNA repair, the nucleotide excision repair. I just want to say that in principle, evolution is related to the fact that we get DNA damage. We can actually produce now artificially very stable DNA molecules artificially, and we see no evolution in them. Paul Chaikin is doing that at NYU. And so the fact that evolution is possible also leads to the fact that DNA gets damaged by various chemical and photo agents. So what is the problem? The problem that I've been interested for quite a while in DNA repair was TT dimers produced by UV radiation. So the skin is exposed to the sun, and the skin is highly regenerative tissue. That is, it cycles frequently because it has to clear those damages. One way to clear those damaged cells, is by just shedding the skin. However, when DNA damage happens, also that DNA damage can be repaired, and then the cell can continue to divide. And it's been observed for a long time, it's been known for a long time that 250 nanometer ultraviolet light creates a TT dimer by basically quenching two pyrimidine, two T bases. And it has been a problem since the 60s, the recognition mechanism of this particular damage. As we've been learning more and more about the repair of these dimers, it became clear that, in fact, it's repaired by this nucleotide excision repair, where once the damage is recognized, new proteins come, they open a bubble. On the damaged strand, about 30 nucleotides are excised, and after that, with ligases, it's actually covered and the correct sequence is restored. And we heard a little bit earlier about how this mechanism works in bacteria. Here I'm going to talk actually about mammalian cells. In the case of this repair mechanism, there are two actually modes that are different in recognition of the initial damage. The first one is during transcription, where a polymerase is stalled. And that stalling of the polymerase actually leads to the recognition that there is something wrong at this point. But there is also the global genome NER, which is actually more mysterious of how the initial signal is being recognized. And what is interesting is that when I started working on this problem, one recognized immediately that, so how is it possible that all sorts of possible damages that can happen, that this NER complex is induced, how is it possible that they're all recognized? Because they're very different from dimers, other damages that are single-strand breaks, and so forth. How could there be a non-specific mechanism that actually specifically recognizes the damage and doesn't try to repair undamaged DNA strands? And using NMR experiments in the 80s, people observed that actually there is a 37-degree kink at the TT dimer. And so it was proposed that this kink is the one that induces is the recognition step in the NER repair pathway. So, but on the other hand, this is a very structural view of DNA repair. And there was no, at that time, any suggestion that perhaps some dynamical changes that happen in the DNA could be also playing a role in the recognition. So, being a physicist and not knowing any biology, I was at that, we decided to set a model, some kind of a model that can, to some extent, reproduce the DNA dynamics. And when I want to say something here about models, the biologist usually criticizes that our models are too simplistic, and this is absolutely true. And actually, that's a good thing, because the very, very complicated model, then we don't know what's happening, because then the computer knows what's going on. So there are, if a certain model captures certain degree of the problem, it could be very useful for letting us think differently, as Steve Jobs used to say. So there is a model that was developed in the late 80s and early 90s, and that model is called the Peyrard-Bishop-Duart model. And that model was developed to try to explain melting curves of DNA. This was one of the problems. And the second problem was to try to simulate solitons in DNA. Now, the solitons completely failed. Solitons are unstable. That was shown by very precise molecular dynamic simulations of DNA. So the solitons story ended around, I guess, 91 or 92. But that model does still reproduce very precisely the melting curves. And around 2004, became clear that the thermodynamic destabilization of the DNA during UV exposure has something to do with the recognition step in in the TT dimer. So this model is a simple model. It has two potentials, one which is between the nucleotides in a single base pair, and then there is the stacking interaction, which is given by the second term. I'm not going to explain here, but for those of you who are interested, probably most of you who actually can understand that, you already know about the model. And so for those who don't understand that, you don't need to know it. So this model can be simulated, as we heard the last few days, using Langevin dynamics. And so we set Langevin dynamic simulations to try to understand, and here is the question that we wanted to understand. What are the changes in the interstrand dynamics when two base pairs are quenched? So this represents the TT dimer. And we wanted to understand what's happening to the interstrand oscillations. So the first thing that we simulate actually was those melting curves. And we actually saw that there is actually shift in the melting temperature. So truly enough, this DNA, so we simulate about 60 base pairs. And we saw that in this 60 base pair, there is a change in melting as the dimer forms. The next thing that we tried was, and then we looked at the interstrand separation, because our hypothesis is that maybe the interstrand separation, if there are very large fluctuations, now because there is a dimer. So I was imagining that, okay, so I have two things that are interacting. And normally when there are no quenching, things are more fluctuating, and they can fluctuate freely. But once you fix something, so I came from condensed matter physics, and I know that you can go through certain phase transitions called, let's say, metal insulator transitions by inducing disorder, which quenches actually the electrons from moving in the lattice. So I was imagining that the same way you can quench these nonlinear waves or nonlinear oscillations that appear in the DNA. And truly enough, at least the molecular dynamics show that actually that is true. And you can see here that two different temperatures. The black and the red curves are with the dimers, and the purple and the pink curve are without the dimers. So we saw those changes. Now, you have to understand that this model that was developed is kind of a very toy model, and that model does not give the real frequencies or time scale of the actual DNA in a cell or even in a fluid. So while this happens at the level of nanoseconds, in fact, we expect that this probably increase is actually happening on a microsecond or maybe tens of nanoseconds. So this story we published in DNA Repair, and we also looked at probability distribution functions, and we looked at where are those fluctuations happening. We found that it's within few base pair around the TT dimer. But what was interesting is that actually the strand that was damaged was not flapping. That was actually quenched. And what was flapping was the undamaged strand. So we proposed that, in fact, the damage is recognized by the healthy strand, which has a different dynamics than the strand would have had if there were no dimer. And we calculated here the probability distribution of bubbles, of those, we call them bubbles, and what is the size of the bubbles. Short-lived bubbles are not changed, but long-lived bubbles actually do have. That means that these oscillations actually change the probability of long-lived oscillation. That's where the large amplitude oscillation appear. And so we did, so that was our proposition. Now, this never happened in biology to me before. I got a call from Hans-Peter Nägele, I don't know if you know him, from University of Zurich. And he said, I want to write together a paper because we just observed that the XPC protein, which is the first recognition step together with DDB2, that actually it binds to the healthy strand. And so we wrote several papers on this subject. It seems that right now it's quite recognized a few years later that actually really it's in the dynamic fashion through the healthy strand, changes in its dynamics, the NER pathway is initiated. Now, nobody has observed yet condensation, and we still think that this is an assembly of number of proteins that slowly accumulate on the break. However, and unfortunately we won't hear that talk by Stoino because he's going to talk tomorrow about something else. But recently there was, they published a very nice Cell paper here from the Institute of Molecular Biology in which they tracked 70 proteins that are involved in DNA damage. And that damage is done with a laser, so there are a lot of damage things that are happening there. There is single-strand breaks, double-strand breaks, but they looked at the foci formation. As the foci forms, it seems that certain proteins are substrates for other proteins that actually could be condensing in a phase separation fashion. So I think that things are more complicated than we imagine at the present moment, because really it's not necessary everything, say, in the cacao bodies to be a condensation. Some things could be solids, other can be actually condensates which are liquid. So it could be actually more complicated. There are substrates over which liquids actually accumulate. So I think that in the next few years and maybe decades, we're going to have very, very interesting physics coming out of those. Whether those are equilibrium, they're probably not non-equilibrium. And it's interesting that in this case, in the NER pathway, there are proteins like RNF168, which are actually being activated by the XPC. So This is in the future will be very interesting to see which proteins condense and which don't, and what is the dynamics. Now, one more thing I would like to mention here that we haven't discussed so far. I haven't seen anyone discuss it. So I don't know how many of you know the Einstein paradox about tea leaves in a teacup. But the problem is that when you stir something, tea leaves get in the center and down rather than go to the periphery. And of course, the solution was given by Einstein, and it's known that it's because the boundaries create pressure towards the center, and that pressure is against the centripetal force. Is it possible, I would like to ask, is it possible that actually DNA dynamics and RNA dynamics and all sorts of other molecules actually lead to this kind of steering mechanisms where actually local concentrations of given proteins are actually increased around damages and around these fluctuating molecules? So it's possible, or maybe it's not possible, I don't know. But I think this is one question that I would like to work on and understand. So in conclusion, I just want to give you that. And maybe it is, we're going to take it out of James Bond, and maybe it is stirred and not shaken in these phase transitions. And, at least because what we don't know is what initiates those transitions. How do, and I want to show you one movie. Well, let me first acknowledge the people that did the work. But I want to show you one movie that unfortunately Cliff Brangwyn couldn't come, but he sent me a movie to show. They recently published a paper in Cell, this was in December, and they were able to engineer the system to show actually the two types of phase transition, nucleation and spinodal formation, spinodal decomposition. So if the movie works, I think you will be amazed. So this is the nucleation. I mean, certainly something's happening, which is very interesting. And then this is their other system. And that's a spinodal decomposition. These are two different types of phase transitions, or two different paths to a different phase. All right, well, thank you very much.
Questions?
Yep.
Thank you very much. It was a wonderful talk. Okay, according to your theory, the strand that is moving is not the one that is damaged, right?
Right.
Which is very interesting. And what about the strand that carry the damage and all those proteins who recognize this damage?
Yeah, so here is what right now we think is happening. The XPC recognizes the flapping on the undamaged strand, but this is very unspecific. Actually, that binding is not very specific and is not very strong. Then XPA comes, and XPA verifies the change in the kink in the 37-degree angle and stabilizes the complex. So this is right now what we think about the NER in mammalian cells. And of course, the rest of the proteins have the role of excising and keeping the whole thing in place. But this is, right now, this is what we think is happening.
Krastan, just
Yes.
one question.
Yeah.
Here,
Where?
Tito. Just one question. You said you did your modeling with a 6-temer of DNA.
Yeah, we changed, we did different.
Or, right. So when you get TT dimers in DNA, at what interval are there? And could that have, obviously, all models are models. I'm just wondering whether the TT dimers were, every 10,000 bases.
No,
and that,
so we did just one single one.
Right.
We didn't do many.
Yeah, but my question is, in real life, when you get a TT dimer, how far apart are they?
Oh, they're next to each other. They're two nucleotides that are next to each other.
That I know, but the next one that you find.
I don't know how far it is.
Okay.
Maybe you can tell.